Hello, AP Bio. Welcome to our video lecture for Chapter 13, the molecular basis of inheritance. So buckle up, kids, because this is going to be a long one. This might have to be divided into two videos. I'm not sure. Chapter 13 is one of the most important chapters in the book. There's a lot of important details, lots of historical stuff to talk about. Plus, it's just a fascinating chapter. Yeah, so this is going to be a deep dive into DNA. So we begin with sort of a picture. It's a screenshot. So a couple years ago, I did one of those cheek swab things. This is through Ancestry.com, where you send it in and they take your DNA and they run it through their database and look at your ethnicity profile or whatever. And this is the one for yours truly. So if you look at this, and if you go to the actual website, it's like when I hover over things, it pops up windows and tells me all kinds of cool stuff. So I am, according to this, 50% England, Wales, and Northwestern Europe, which is the yellow circle or oval right here. 18% Germanic Europe, which is here. 16% Ireland and Scotland. How interesting. 6% Sweden, 5% Norway. That's Norway. That's Sweden, right? Yep. 5% France, just for some flavor. And then the website also does this thing where they have whatever database to track the migration of your family. Like in this case, from the old world to the new world. So, you look at this and you're like, is that really accurate? Like, do they really know what they're talking about? Well, let me give you a little history lesson about the Kuhn family. I know you've been dying to hear this. So let's go back, 1844, all right? Johann Heinrich Kuhn, whose name got anglicized. Is that a word, anglicized? It got changed to the English version of John Henry Kuhn. 1844, he is 6, I believe. And he immigrates with his parents, who is also Heinrich Kuhn, and his wife, Regula. That's a great name. From a town called Düsseldorf, Switzerland, which is in the northern part of Switzerland. In a German-speaking part of Switzerland. Kuhn was spelled K-U-H-N. It got changed to K-O-O-N. We have census records showing all this, so I'm not making this up. Düsseldorf, Switzerland is right about there on the map, which is right smack dab toward in the middle of where all these circles overlap. So it's, now that's just the Kuhn line. Obviously, that's, many great-great-grandfathers before you get to me. The migration story of my family. So the Kuhns migrated to an area which is now under Lake Murray, which is near Chapin, South Carolina, which is where I was born, which is right here, right? That's actually maybe to the left-hand side of that dark circle. My mother's family originated in Alabama, which obviously is going to be right there. Her last name was Keating before she got married. So looking at this, like when I saw the DNA results, I'm like, holy cow, this is like, all the things I was told as a kid were actually true. The DNA shows it. Very interesting. Okay, so before we get into, the story of DNA, I want just to stop for a minute and just pause and think about what DNA actually is, all right? Forget about molecules, forget about biology, just what is DNA? We talk about DNA as a code, and it is a code. It's a code for life, but I think of it more as a program. So think about what like an acorn is. An acorn is a little seed for an oak tree, right? If you take an acorn and just throw it outside, all right, all that acorn needs to grow into a massive oak tree is water, sunlight, some soil, and the micronutrients in the soil. That's really all that it needs. All right, all the programming is there. Think about if I asked you or as a computer scientist to design a computer the size of an acorn, or I could just throw it outside and it would grow into a computer the size of an oak tree, and you really had to do nothing to it. And that it could then reproduce tiny little computers that could grow into other computer-sized oak trees. That's insane. Think about the programming that you would have to do to make that happen. Well, nature did it for us. Like DNA is basically a code for a program that creates life, right? Our computer programs are based upon a digital code, like ones and zeros, right? Binary code. The biological code is based upon, you could think of it as the numbers 0, 1, 2, and 3. There's 4 parts to the code. The 4 molecules, A, T, G, and C, adenine, thymine, cytosine, and guanine. But basically all that life is it's a code. It's not a digital code, it's an organic code using molecules. All right. And it's a code not only that will grow into an organism, but it also has the code for how to make more of the organism, which obviously we don't have self-replicating computers yet. Maybe one day we will. It's a little scary actually. But we're basically just a biological code. All right, that's what DNA is. It's the instructions for creating you, and there's, it's a built-in code in a molecule, which we call DNA. Now, you've heard about the guys Watson and Crick. There's a picture, that's Watson, that's Crick. We're gonna get more into them in a minute. They did not discover DNA, they discovered the structure of DNA. People knew about DNA going back to the 1800s. They knew that there was this stuff inside the nucleus that was slightly acidic, so they called it nucleic acid. They didn't know what it did though until later in the 20th century. So Watson and Crick, very, very famous guys. They get the Nobel Prize, I think in 1962, it's the early 60s for sure. In 1953, they publish a paper. It's a very short paper in a journal called Nature. It's a very famous journal where they proposed a 3D model of DNA, which is the double helix. They were correct. Obviously, they got the Nobel Prize. So they get credit for discovering the structure of DNA. Kids always get this confused. They didn't discover DNA, they discovered the structure. And once you know the structure, obviously you can do stuff with it. This picture is taken, this is Watson. I think Watson's in his 20s at this time, and I think Crick is in his 30s. So they're two youngsters. This is in England. This is a big 3D model they had actually built. And we're going to come back to the story of the discovery of the double helix later, because there's more than two people involved. Yeah. Okay, so let's go back a second. So mid 20th century, people want to know, like, how is the code for life carried? And they know that it's carried by chromosomes. They know that. People know about mitosis. Well, chromosomes are made of DNA. They're also made of protein. Remember, we know that the protein part of DNA is just for structure. The histone proteins, you wrap the DNA around, but they want to know which molecule actually codes for inheritance. Well, they know that proteins are enzymes, or enzymes are proteins, which are very useful in cells. And they know that proteins have a code of 20 letters, which we call amino acids. So the going, the best bet for what molecule codes for life is that it's proteins, because there's already, our alphabet has 26 letters, right? Well, proteins have 20 amino acids. So that makes sense, where that code would be good enough to turn an acorn into an oak tree. Of course, we know it's wrong. Proteins don't code for life. DNA codes for life. They knew that DNA had 4 nucleic acids. Had 4 nucleotides, A, T, G, and C, which they figured was not enough variety to code for life. All right. Couple famous experiments. You don't need to have these experiments memorized by any means. The AP exam might give them to you to analyze. So let's just go through them briefly. Frederick Griffith was a scientist in the 20s. He worked with pneumonia, which is a bacteria, in mice. And this slide explains what he did. I want to go through it with a diagram. So there's a strain called the S strain of pneumonia. You inject it into mice and the mice die, okay? There's a strain called the R strain. You inject it into mice and the mouse does not die, all right? If you take S and you heat it, you boil it, you kill the bacteria, right? You inject that into the mouse is fine because you killed the bacteria. This last one's the interesting one. So he would take S that have been heat killed, so they're dead, and R that were living. The mouse should be fine, right? Because you killed the S strain. We'll inject the combo. Not only does the mouse die, but inside the mouse, you find living S. So the S came back to life. So what's happening is some molecule is being transferred from the heat-killed S to the R. And, hint, the molecule is going to be DNA. But the term for that is called transformation. You move DNA from one cell to another. Okay? Now, another experiment. Alfred Hershey and Martha Chase. They experimented in the 50s. With viruses called bacteriophages. A bacteriophage is just a virus. The name is misleading. It's a virus that infects bacteria. So viruses, we're going to come back to in a later, but viruses basically have 2 components. There's the outer protein coat, like look at a virus. Here are viruses. These are bacteriophages. Nice geometric shapes. So the protein coat's on the outside, and on the inside, there's some nucleic acid, either DNA or RNA. That's all that a virus is, all right? And viruses infect living cells and take them over and create more viruses. So the question is, which molecule does the taking over? Is it the protein coat or the nucleic acid inside? Well, Martha and Chase were very, very smart. Hershey and Chase were very, very smart. They knew that proteins contain the element sulfur. Nucleic acids usually do not, all right? Nucleic acids contain the molecule phosphorus. Proteins usually do not. So they grew a batch of viruses in radioactive sulfur, and then a batch in radioactive phosphorus. And you can tell where the molecules go by detecting the radioactivity. The way a virus works is, here's the protein code, then they inject their DNA into a cell, and hint, the DNA takes over the cell. So what they would do, and to do this, they just used a blender. It's a very fancy experiment once you have the bacteria, or the bacteriophage is grown. And so here's what happened when you infected the bacteria with the radioactive sulfur bacteria. You inject them, when you put them they infect the cells, put them in a blender, the little protein coat gets kicked off and the DNA stays inside, right? And then you have billions of cells that are infected. Then you centrifuge the sample, and the cellular part forms a pellet in the bottom, and the liquid part is on top of the pellet. And the sloughed off protein coats are in the liquid part because they're not as heavy as the cells. All right. Then do the same thing with the radioactive phosphorus. All right. And the key is find where the radioactivity is. If the radioactivity is in the liquid part, that's not inside the cells. If it's in the pellet, it's inside the cells. And long story short, when you did the phosphorus that was radioactive, the pellets were radioactive, which means that's what actually goes inside the cell to infect it. So this shows that DNA, which has radioactive phosphorus, remember the sugar phosphate backbone, is the molecule that's doing the work of inheritance. In this case, doing the work of infecting cells to make more viruses. Okay. This slide just goes through the experiment. You can read this yourself. It's what I just said. Erwin Chargaff, this is a guy worth noting. He's the guy that figured out the base pairing rules. Well, actually, he didn't figure out the rules. He figured out that like the numbers of A's, T's, G's, and C's from one species to another vary, actually from one individual to another, really. But he figured out that in any sample, the amount of A equals the amount of T, the amount of G equals the amount of C, which we know today is the base pairing rules. So this chart shows some sample data. So in a sea urchin, 32.8% A, 32.1% T, 17.7% G, 17.3% C. Like, look at the numbers. A and T are always the same. We're going to round. And G and C are always the same. And that's not a coincidence, right? Like, there's a reason for that. We know it's the base pairing rules. A common multiple choice question is, if I give you one letter, can you tell me the percentage of the other letter? So if I say, for example, that a sample is 15% A, right? What percent would be T? Well, the answer is 15% because they had to be the same. Well, if it's 15% A, can you tell me the percent G, right? Think about that for a second. If it's 15% A, that means it's 15% T, which is 30%. So what's left? 70%. Cut that in two, the answer is 35%. Let's do another one. If I tell you that a sample is 40% G, how much A? So if it's 40% G, that means it's 40% C, which is 80. There's 20 left. So that means there's 10% A, 10% T. All right. Make sure that you can do those. The math isn't hard. You just have to kind of go through the logic in your head. So this shows a single strand of DNA. We did this in chapter 3. So there's the sugar phosphate backbone with the phosphate groups, the five-membered ring. In this case, it's deoxyribose. If it's RNA, it's ribose. And then your nitrogen bases, T, A, C, and G. Notice that T and C are a single ring, and A and G are two rings fused together. That's going to be important later. This just shows a nucleotide. We did this in chapter 3. Notice the carbons are labeled 1', 2', 3', 4', 5'. Okay, so let's get to the story. This gets to the interesting stuff. So James Watson, Francis Crick, Marius Wilkins, and Rosalind Franklin. So let me skip ahead to the end for a second. So in 1960, I think it's 62, when Watson and Crick get the Nobel Prize for this, Watson got it, Crick got it, Marius Wilkins got it, Rosalind Franklin did not get it. And don't be too outraged. The reason she didn't get it was because she was dead. And the Nobel Prize rules, you can't give it to someone who's died, which to me is kind of a silly rule because, you know, that's, I don't know, I don't love that rule, but you can't give it posthumously. And she died of ovarian cancer. How she got that, we'll talk about in a minute. So some quick backstory. James Watson and Francis Crick are working at Cambridge University, which is in England, in a lab called the Cavendish Lab. Maurice Wilkins and Rosalind Franklin are working at nearby King's College, which is also in Cambridge. James Watson was American. Francis Crick and Maurice Wilkins were British. Rosalind Franklin was also British. She actually had been born into a very prominent Jewish family in London. They were involved in resettling some Jewish refugees during World War II, and actually took in 2 Jewish children into their own home, which is pretty cool. She had kind of made a name for herself in Paris doing this technique called X-ray crystallography. And X-ray crystallography is a technique where you do it to determine an object's structure, and you take a sample and you crystallize it, which is kind of hard to do sometimes, and you shoot X-rays at it. And then the X-rays like interact with the crystalline structure, and behind it, you have an X-ray film, and you see how the X-rays develop on the film. It's sort of like shining light and analyzing something's reflection. And you use the X-ray image to figure out what the structure is. And there's a lot of fancy math and trig that you use to do this, but getting the images themselves is tricky. And when she was in Paris, she became pretty famous for doing X-ray images of carbon-containing compounds, particularly coal and graphite, and she figured out the structures of things through their X-ray images. And she was, at this point, was possibly the best person in the world at doing X-ray crystallography. All right, she gets offered a position at King's College in London, and she takes it. And she gets there, and she is working with this guy, Maurice Wilkins. All right, so it's Watson and Crick at Cambridge, and Wilkins and Franklin at King's College. Franklin and Wilkins did not get along. James Watson wrote a book called The Double Helix. I've never read it, but I've read things that it talks about, different accounts of how it was discovered. And Wilkins and Franklin, I mean, it was so bad that Franklin ends up leaving and going to a different university. Wilkins is described as being very, very shy, very soft-spoken. And Franklin's described as being very outspoken. And like the book describes it, like when she was talking to you, she would like stare at you in this way that like made you feel uncomfortable. It's probably overblowing it. But like Wilkins, he did not like Franklin. He thought she was too forward, and they just did not get along. To make matters worse, I think his last name was Randall. The guy who ran the lab that Franklin and Wilkins worked in, when Franklin came, Wilkins thought Franklin was working for him, when in actuality they weren't, like she wasn't under him. But the supervisor did not make that clear. So they just did not get along. There were power structures going on. There's actually a story where later, let me get this straight, it's either Watson or Crick go to King's College to talk to the two of them, and Wilkins isn't there, so they go talk to Franklin, and she gets mad about something, and like Frank or Watson like steps backwards and bumps into Wilkins and knocks him over or something. Whatever. It's whatever. These two did not get along. Okay. So I'm going off too long, but the story is it's interesting, and it doesn't end there. So Franklin creates this very famous X-ray image of DNA. It's right here. It's called image 51 or X-ray 51. It's very, very famous. It's the best X-ray picture of DNA that's ever been created up to this time. She's planning to leave King's College because she's not happy there. And any work that she has done there stays there. It doesn't belong to her, it belongs to the university, right? So one day, Watson and Crick come over, and Maurice Wilkins shows Watson and Crick this image without Franklin knowing about it, which, you know, people kind of vilify them for that. But, you know, the image didn't belong to her. So there's debate as to who is really at fault, if there's even any involvement in the first place. Without her image, they wouldn't have figured out this. They wouldn't have been the first ones to figure it out. And they, in their article, they give her credit. They mention her at the end, her and Wilkins, that their contributions helped them figure it out. Franklin had already figured out that the backbone, I had this somewhere, that the backbone, where do I have this? She figured out that the double backbone was already sugar and phosphate. There was one model that had the sugar phosphate thing in the middle, like a central thing with the nitrogen bases coming out like spokes of a wheel. That's just wrong. Like that's flat out wrong. She figured out that they were on the outside, and she, you know, people pretty much agree that she would have figured it out all by herself eventually. They used her work to get to it first. Now, there's more to the story. So like when Watson and Crick publish in Nature, the publisher also puts articles by Wilkins and Franklin in there also after Watson and Crick, showing that their data supported their model. And, you know, Franklin saw that. She'd already gone to a different university. And she wasn't convinced Watson and Crick were right in their model because they didn't have enough evidence to fully prove it. And she didn't want to publish till she could prove it 100%. And Watson and Crick were like, well, I mean, it's mostly right. So, you know, they went ahead and published it, and it ended up being 100% right. So she was very hesitant to publish. So like, you can't really blame Watson and Crick. There's no real villain of the story. In the late 50s, she gets ovarian cancer and dies. And she probably got ovarian cancer due to exposure from x-rays. So her work literally killed her, more than likely. She doesn't get the Nobel Prize because they don't award them posthumously. She likely would have gotten it if she wasn't dead. But in the story of DNA, you hear Watson and Crick. You don't hear Wilkins and Franklin much, especially Wilkins, to be honest. Franklin, you hear a lot more because people want to, you know, push her back up where she belongs. And a lot of the problems, like Franklin got along with Watson and Crick fine. She vacationed with Crick's family in the late 50s. And when she got sick, she actually spent some time living with them or vacationing with them. Wilkins is the one that she really did not get along with. Yeah, so there's lots of complexities to the story. It's just fascinating. Okay, so let's go back. So what is the model? So it's a double helix. There's the sugar phosphate backbone. The rungs, the G and C and A and T are in the middle. Again, I haven't read the double helix, the book. I should probably read it, but it's, you know, it's written by Watson, and it recounts all of this. And, you know, there's some charges of sexism in there. Of course, we're talking about the 50s, so that's not that surprising, although it wouldn't be surprising today, to be honest. Okay, so here's a nice textbook image showing the sugar phosphate backbone with G and C and A and T. We've seen these before. This just shows the same thing. Here it's wound, here it's stretched out. This is called the space filling model. Notice we pointed out in chapter 3 that the two sides of DNA are held together by hydrogen bonds, which are pretty weak. Notice in the picture between A and T, there's 2 hydrogen bonds, and between G and C, there's 3. Just notice that. If I wanted to break these bonds, how could I do that? You just heat it. If you heat DNA, the two halves fall apart. If you cool it down, they'll come back together. Remember, heating hydrogen bonds breaks them. This just shows some measurements to 3.4 nanometers. This is what Franklin wanted to have all this figured out using the X-ray images before she published. Watson and Crick were ready to go ahead and publish without having all the numbers fully figured out. And, you know, the early bird gets the worm. The first one that publishes are the people that get credit. This is the same image on the previous slide, the same image. So Watson and Crick also figured out that the 2 strands were anti-parallel. Remember we discussed in chapter 3 the whole 5' to 3' deal, that one strand, let me go back. This strand is going 3' to 5' that way. This one's going 5' to 3' that way. You see how here the O's point up and here the O's point down? That's called anti-parallel. The 2 big bases are called purines. The small ones are called pyrimidines. They figure out that, you know, to have uniform spacing, so the helix doesn't bow out or get pushed in, you have to have 1 purine and 1 pyrimidine, so an A and a T or a G and a C. So they use Chargaff's rules to figure that out. Yep. They also figure out how DNA replicates. So the way DNA replicates is you have a strand of DNA, the strand separates, and basically, like, to build a new strand, so here's an A, you bring in a T. Here's a C, bring in a G. Here's a T, bring in an A. So you take the parent strand, you separate it, and then you build new strands using the originals as a template. So this new DNA helix is part original strand, part new strand. The word for that is semi-conservative replication. Competing model, so conservative would be you have a parent and you copy it. The original strand is there and the new strand is all brand new material. The semi-conservative model, which is what really happens, you have the original strand of the two new strands, half of it's original, half of it's new. This dispersive model is each strand is a mix. This is incorrect. B is the correct way that DNA replicates. And Watson and Crick figured that out too. Okay, so at this point we need to go through some detailed. We gotta the devil's in the detail, and here comes the devil. Sorry, that didn't come out right. You get what I mean. So as the enzymes involved in DNA, replication. Okay, so let's just go through it. So look at the picture. So DNA replicates at what's called an origin of replication, and that is the spot where an enzyme called DNA helicase comes and takes the strand and separates it, right? You end up with a thing called a replication fork. So here's the molecule helicase. It is unzipping the DNA strand going to the left, all right? Notice that, okay? Topoisomerase, so if I took a string, like a piece of thread, and I started to like unwind it, it gets tangled downstream. Topoisomerase is an enzyme that helps stabilize the structure so it doesn't supercoil. I just use the term downstream. So helicase is going that way, right? Downstream would be going, that way, the direction of the current. Upstream is up here. So in this picture, topoisomerase is downstream of helicase, and primase is upstream. Okay. So helicases unzip DNA. Single-stranded binding proteins, or those are upstream of the helicase, and they stabilize the structure. Topoisomerase stabilizes it downstream. Now, DNA polymerase, that isn't on this slide yet. Polymerase is the enzyme that actually builds the new strand. So helicase unzips, single-stranded binding proteins, topoisomerase stabilize, polymerase builds. Okay. With bacteria, so remember bacteria only have a single circular chromosome. Like, look at the picture. You start unzipping the helix at this point, and once you do that, you can start replicating in both directions. And by the end, you have a new strand or a new circle of DNA. Our chromosomes are linear. They're long strings of DNA. And what I can do here is I can start to unzip in more than one place and then copy it going in both directions. So like, you don't copy a chromosome from beginning to end. You copy it at multiple sites simultaneously, and by the end, you have a new copy. Okay, so here's a theme for you. DNA polymerase, and there's more than one type. That's why it says polymerases, is a brilliant molecule. It's an enzyme. Without it, you can't copy DNA. You gotta have it. But DNA polymerase it has some design flaws. It was kind of like when we did photosynthesis, and we said that Rubisco has some design flaws in it. It has some shortcomings. Remember how we said how, evolution doesn't create from scratch. Evolution takes what already exists and tries to make it better, like updating your phone's operating system. You don't get a brand new code, you just patch the one that you have. So polymerase has some problems. And because it has some problems, this process gets very complicated because we have to have fixes. The first problem we're going to discuss that polymerase has is it cannot start a strand from scratch. It cannot bring in that very first nucleotide. It doesn't, the active site, it can't bring in a nucleotide to start it. So you have to create what's called a short RNA primer. RNA primers are made of RNA. They're single-stranded. They're 5 to 10 nucleotides long. The enzyme primase makes the primer. And guess what? Primase can start from scratch at the beginning. So primase comes in, puts in the first 5 or 10 bases made of RNA, which is going to be a problem because it's going to have to change that to DNA later, right? And once you've primed the new strand, then polymerase can come in and pick up where primase left off. So because polymerase can't start the strand from scratch, you have to bring in primers and primase, which is an extra level of complexity. But nature's complicated because polymerase can't do it all at once. It has design flaws. So primase comes in. Okay, now the way primase works, remember how carbon had the 1', 2', 3'? The 3' carbon is, look at this picture. It's that one right there. That's a 3' carbon. See, it has an OH group. Polymerase can add a new nucleotide to a free 3' OH. So in this picture, polymerase can add a base there. It cannot add a base up here. It adds to a free 3' OH, all right? Which is another design flaw because it means polymerase can only go in one direction. Fun factoid, polymerase can bring in about 500 nucleotides per second in bacteria, which is super fast, about 50 per second in human cells, which is still super fast. And notice, like helicase is going that way. Polymerase isn't in this diagram, but it would start where the primase left off, and it would be going this way too. So that 3' as a free 3' OH group, polymerase can start adding new nucleotides going that way. Polymerase could not start adding nucleotides going that way because that's the 5' end. All right, so that's a problem. We're going to have to add more complexity to fix that problem. What else do you? So you need A's, T's, G's, and C's, right? So you have ATP. You also have GTP, CTP, and TTP. The cell makes those two. We haven't discussed them yet, but the cell does. The D stands for deoxyribose. So D-ATP is deoxyribose adenosine triphosphate. Those molecules give you energy, which you need, and they also give you A's, T's, G's, and C's. Okay. Okay, so big problem. Let me show you this. Which diagram do I want? Let's look at this diagram and then go back to the words. So I have two origin of replications. I have this one, and this one started, helicase is going that way. This primer up top had a free 3' OH. Polymerase started here, and it's building a new strand going in the same direction as helicase, not a problem. This works great. Well, what about the bottom half, right? The bottom half, going back in time, helicase was here. Helicase is going that way, but this strand has to go to the right direction, right? Towards the right. So once helicase unzips it to here, you got to have a new primer. When it gets there, there's a new primer. That's called the lagging strand. So the leading strand, one primer does it. The lagging strand's being unwound in the wrong direction. And on the other side, they're opposite, right? Because here helicase is going that way. These fragments are called Okazaki fragments. Named for a guy whose last name was Okazaki. And this adds extra complexity. Plus, wherever you have a primer, you're going to have to later kick out the primer and put in DNA instead of RNA. So because polymerase is a directional enzyme, I have to add more players to the story to fix that, all right? Remember, evolution could go back and just redesign polymerase from scratch, but that's not what happens. You take what you have and you make it better. You don't start from scratch. This slide just uses words to explain what I just said. This slide shows, so helicase is going that way. The leading strand goes that way, not a problem. We've already discussed this slide. This term, the enzyme DNA ligase, let me actually look at this one for a second. In this diagram, helicase is going this way. On the bottom, polymerase is going that way. So I had to synthesize the new strand in fragments. Now, polymerase is still an awesome enzyme. How do I get the primer off and bring in DNA? Guess what? Polymerase can do that because it has free 3' ends at this point. Polymerase basically comes and reads the strand again. It can proofread itself, which is a great little tool that it can do. It can kick off the RNA and bring in the DNA. The last thing you have to do is, so like this DNA polymerase, in this case is polymerase 3, don't worry about that, is going to come and bring in the new strand. So you're going to, like right here where the number 4 is, I have to link the blue with the next strand of blue. Actually, not in 4. 4 is where, like say polymerase comes and gets rid of all the red, like to link up the new blue with that blue strand, what kind of bond is that? Well, that's part of the sugar phosphate backbone, right? So those are a covalent bond. So I need to covalently bond when this strand becomes blue to the blue that was already there. The enzyme that does that is called DNA ligase. Ligase is the last enzyme that comes along, and wherever it needs to add a covalent bond, like if here's a piece of DNA, here's a piece of DNA, I want to link them together. These aren't base pairs. These aren't like the same part of the helix. This is two that you want to link horizontally. Ligase comes and links those together. So because polymerase isn't perfect, you have to have primase and ligase and the fragments and the primers and all that story for this to work, all right? The book has all these great diagrams that go through all this. You know, at some point look through these diagrams, but it's what we've just been talking about. This is from a different textbook, but this just shows the whole story. Okay. Now I've already said this, but let's just say it again. Polymerases not only copy DNA, but they can proofread themselves. Nucleotide excision repair is this deal where, so say I have a T and you should bring an A, right? Say you brought in a G by accident. Usually the helix or the strand kind of bows out, and enzymes can come in and cut out the base that's wrong, and polymerase will come in and bring in the base that's correct. So, you know, polymerase makes mistakes, and a mistake in your DNA is called a mutation, right? Well, polymerase can also fix the mistakes that it makes. And it has a rate, so say polymerase can fix 10 mistakes a second, all right? If you get a sunburn, and in that second your DNA had 20 mistakes in it, those mistakes become permanent. If your DNA mutates faster than polymerase can fix it, that's a problem, and that could lead to cancer later, right? That's what this slide says. The error rate, you know, is low, but it's not zero. Mistakes in the sequence that don't get fixed by the time the DNA gets copied again become permanent, and they can't be fixed. Another problem, so DNA goes, you know, DNA goes 5' to 3'. You know, because you need a free 3' OH, the first one you bring in is the 5' end. So it goes 5' to 3'. It's always 5 to 3, 5 to 3, 5 to 3, 5 to 3. The new strand is 5 to 3. So what about the end of a chromosome that ends with a free 5' end, right? You can't bring in that last nucleotide. And you know, your cells copy 1 billion times or whatever. So as a result, as you get older, your chromosomes actually get shorter because you can't complete the 5' end, which is a problem. See in this picture, the chromosomes are shorter. So you have an enzyme called, and by the way, the ends of chromosomes are called telomeres or telomeres. Telomerase is an enzyme that will lengthen chromosomes in egg or sperm cells. Because if this happens in an egg or a sperm cell, you know, those are called germ cells. I'm not sure I've said before. Germ cells are what you call egg and sperm cells, like germinate. If a mistake happens in an egg or sperm, they're going to be in the zygote. The whole organism will have the mistake. So egg and sperm cells have telomerase to lengthen their chromosomes. They just add extra bases. It's like adding a sequence that if you screw it up, it doesn't matter. It's like a gray area you could cut off if you needed to. Some people think telomerase might be some kind of cancer drug. I'm not sure where that stands right now. But your chromosomes do get shorter as you get older. So some people relate that to the aging process. Anyway, you need to know what telomeres and what telomerase does. Chromosome structure. So we've discussed how in bacteria it's a single circle. This picture looks like a rubber band that you got kinked up really tightly. If you unwound it, it would be a circle. The nucleoid, remember, our bacteria don't have nuclei. They're just in a region called the DNA is in a region called the nucleoid region. This is shown E. coli. Your chromosome or your DNA is organized into chromosomes. We've already said this. Chromatin is the mix of DNA and protein. You see this picture, the DNA is the blue wound around the purple histone proteins. You take that, wind it up really tightly, and then you form what are called chromosomes. They're linear molecules, right? So heterochromatin and euchromatin. So DNA can be wildly like compressed, tightly packed, or it can be loosely unpacked. If it's unpacked, you call that euchromatin. If it's tightly packed, you call it heterochromatin. DNA that is tightly packed is not expressed, right? So if I want to turn a gene off, you just wind it up really tightly. Euchromatin, you know, the root EU stands for good or true. Euchromatin is expressed because it's unwound. So during mitosis, when your DNA is tightly packed, that's heterochromatin. During interphase, when it's unpacked, that's called euchromatin. And there's ways of doing this that we'll talk about later. Here you just show how the DNA gets wrapped around the histone proteins, and you form the little loops, and they form back on themselves and all that. Then you make a chromosome. Okay. All right, now, so that's a lot. So we're not done yet. Switching gears. So let's skip ahead to DNA technology, all right, and different techniques that we use in the lab with DNA. So you know that DNA has the base pair rules, right? If I gave you a strand of DNA that was T, 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 and said, what's the strand next to it? What's the complementary strand? It would be a bunch of A's repeating, right? If it's G's, it's C's. Nucleic acid hybridization is basically like I can go in a lab and make a DNA sequence. I can make one that says GGG, AAA, TTT, TCAGT, and then it will bond. The hydrogen bonds will stick to the complementary sequence due to nucleic acid hybridization. So if I want to find where a gene is on a chromosome, make the complementary sequence, make it radioactive, put it in the test tube and see where it sticks. And that's where the gene is. It's almost like a heat-seeking probe, but it's seeking the complementary sequence. That's called genetic engineering because, again, we're engineering genes. So let me tell you a different story before we get into different techniques. So if you have diabetes, right, you probably take insulin. Insulin is a drug that if you take insulin shots, that insulin is human insulin, right? But insulin, I don't, by no means do I want to say that it's cheap because it's getting more expensive, but insulin is not something that's so expensive no one can afford it, right? So back in the day, if you were diabetic, you probably died from it. And the only way to get insulin was to get insulin from a cow. And what they would do is they would either, well, you could get it from a cow or from a dead body. From a cow, you could take the cow's pancreas, which makes insulin, and grind it up and get like a little bit of what's called bovine cow insulin. Obviously, you know, you're going to be killing the cow, which, you know, you do this from people who are making beef anyway. And bovine insulin, you know, it worked okay, but it's not human insulin, so it doesn't work quite as effectively as the real thing. Or you can take a cadaver, a dead person, grind up their pancreas, get some human insulin. That works great, but you know your source is going to be running out because you have to use dead bodies, right? So insulin was expensive. It wasn't widely available. Problem. Well, today insulin's widely available. It's human insulin. So what happened? Well, the story goes, and I think was this the mid 20th century, maybe going into the 70s. I'm not sure when it was done. You can Google it yourself. So some scientists, the company was Eli Lilly, which is a famous drug company. They found where the gene for insulin is in a human. Say here's a chromosome, the gene's right here on chromosome number 10. And they used enzymes and they cut out the gene for insulin. And then they put that gene into a bacteria, right? And the bacteria started producing human insulin. Now, the story is actually more complicated than that because there's a lot of things you have to do to get the bacteria to take the DNA, to get it to work right. The enzymes aren't quite the same, so it actually was a very difficult thing to do. But once you have one bacteria that can make insulin, that bacteria can be 1 billion bacteria in a day. Now you have huge vats. It's almost like going into a brewery that makes beer. You have huge vats full of bacteria that are making human insulin as long as you feed them. So you feed them sugars or whatever, again, like you're making beer, and they're just pumping out human insulin. So it's a human product that's made by a bacteria, which are cheap and easy to breed, right, or to grow. So the question is, how do you do all that, right? The term recombinant DNA, see it's the word recombine, is basically any strategy, any technique where you're combining DNA from different species together. The term plasmid, so remember bacteria have the one circular chromosome. They also have short little, these little like rubber band looking things, short little circles of DNA. They could be just a couple thousand bases long. They can have dozens, if not hundreds of little plasmids in addition to their big circular chromosome. What I want to do is to take a bacterial plasmid, take it out, put a human gene into it, and then give the plasmid to the bacteria. That's what I want to do. And once you do that, you do it through a process called gene cloning, where you put the gene into a bacteria and then grow it. So you're making copies, so it's like a clone. All right. This picture from your book. So this is showing it for, you know, we discussed insulin. This is showing how you do it for human growth hormone, for any gene you want the bacteria to express, you do this. So look at the diagram. Here's the bacterial chromosome. I don't care about that. Here's the plasmid. I take it out. The black part of the plasmid, I put the gene of interest into it, give it to the bacteria, and hope, excuse me, that the bacteria express that gene. And if they do, they just pump out whatever product the gene makes. Now, okay, I keep saying how you're cutting out DNA. How do you cut out DNA? So bacteria naturally make these things. We're gonna do a lab that involves this later, so we're gonna revisit this. These enzymes called restriction enzymes, bacteria make these naturally. They're sort of like a gift that nature has provided us because we use these in biotechnology. These are enzymes that cut DNA at specific sequences. Look at the picture. So you see how this DNA is cut? It's cut like a blunt cut, then a cut through the hydrogen bonds, then another blunt cut through the covalent bonds. And the ends are called sticky because the base is kind of overhang, and this AATT would rebind with this TTAA, right? So these are enzymes that cut DNA at the staggered type cut at specific sequences in really anything. It doesn't have to be a bacteria. Why bacteria make these enzymes? It's actually a defense against viral infection, which we'll talk about that later. There's a great reason why they make them. We use them for our own purposes. So say, like, look at this diagram. So the green is the plasmid DNA. Say that I cut it with an enzyme that left me with a TTAA overhang. So this gray gene is, say that's from a human. I cut it with the same enzyme. So the sticky ends are the same. So put them in a test tube. Guess what they're going to do? They're going to bond. The hydrogen bonds will come together, and you'll form a new piece of DNA where you have the plasmid with a gene in the middle. Now, is it possible that the two green ends could just come back together without the gray gene in there? Yes, it is. But some of the cases are going to involve the gray coming in sticky ending. That's not a word, but I made it up. Sticky ending. The ends will come together to form the new piece of DNA. And all I do now is I add DNA ligase. Ligase was the enzyme that would link up a covalent bond there and one there, and suddenly you have a new recombinant plasmid. Okay, great. So when we do, actually the lab that we're going to do is on gel electrophoresis, which is what we're going to talk about right now. Electrophoresis is how you separate DNA fragments. Okay, so we're going to do this in class, so let's just go through this briefly. So let me look at a different picture. No, let's do it now. So you cut up DNA into fragments. The fragments are different sizes. How do you separate things based upon their size? So gel electrophoresis, like this is an actual picture. Agarose gel is made from seaweed. You'll see this in class. And inside the gel are sort of like microscopic threads. And the threads are kind of like this number, where there's gaps between the threads, and the gaps can be big or they can be small. It's sort of like using a strainer if you cook pasta noodles. And if I can get the DNA to move through the gel, smaller fragments move faster because they get through the gaps, right? Bigger fragments get hung up in the threads and they move slower. So what I do is I put the DNA into these little wells in the gel, and I want the DNA to move down the gel. Well, how do I get DNA to move? Well, there's many ways you could do it. You could just let gravity do it, but that's going to be pretty lame. I'm going to get the DNA to move due to an electrical current. So remind yourself back from chapter 3. Does DNA have a charge? And if so, what is the charge on DNA? All right, the answer is DNA has a negative charge because the phosphate group, the phosphate sugar backbone are negatively charged. So if I put DNA into an electric field, DNA will move toward the positive end. And here I had this set up to a power source. Again, we're actually going to do this in class later, where here's the cathode, here's the anode. The DNA is going to move toward the positive end, and as it moves, smaller fragments go faster. Here, the fragments are stained with a dye that's glowing under UV light, so you can visualize them. Like in that fragment, so the welds are up here, the DNA started up here. That fragment is smaller than that fragment. So if you know what size the fragment should be, you know which fragment's which. Yeah, okay, another technique. So PCR. So, let's say there's a murder and you find one hair from the murderer at the crime scene. That's got DNA in it, but it's not a whole lot of DNA. If we can have a way of making many, many copies of a piece of DNA very, very fast, that would be fantastic. We had this technique called PCR, called polymerase chain reaction. This is actually invented by a guy named Kary Mullis. He invented this, and he got the Nobel Prize for it. And he was born in North Carolina, but he grew up in South Carolina. He went to Dreher High School, I believe, in Columbia. He passed away in 2019, I believe. But yeah, he got the Nobel Prize. He's a very interesting character. There's some heavy drug use involved there, which in class we'll talk about later. He's out there, but he invented PCR and got the Nobel Prize. So, yay. So if you look at the diagram, what happens is, so could I do replication of DNA in a test tube? What would I need? I need the DNA. I need helicase. I need polymerase. I need A's, T's, G's, and C's. I need the primer. I need the primase. Could I do all that in a test tube? And the answer is yes, yes, you can. The problem is I can't just throw in helicase because, actually, I'm not sure why you can't throw it in there, but I guess because it just unzips everything. So just adding helicase actually doesn't work. I have to separate the strand by heating it. So look at this diagram. So in cycle 1, I heat the strand, it separates it, and then your primers and your polymerase and your primase come in and build, actually I'm not sure you even need primase, builds the new strand, then you cool it down, then you heat it again, and each new strand makes a new strand. So every phase of the cycle is exponential. So 1 helix becomes 2, becomes 4, becomes 8, you get the idea. So after a couple hours, 1 strand of DNA can become a bunch of DNA super fast. PCR is a way of copying DNA, which is super important. Though one of the tests for coronavirus, you might hear it on the radio, they call it the PCR test. What they're doing is they're detecting the coronavirus by detecting in this case, it's actually RNA, I don't think it's DNA. But you're looking for the RNA of the coronavirus. If you detect RNA from the coronavirus, like that means you have coronavirus, right? It's called the PCR test because you do PCR to copy the sample really fast so that you can detect the right DNA. If you only have one piece of it, you're not gonna be able to find that. You need to have a bunch of pieces of it. So like you're copying the coronavirus DNA to see if you have it. Now, problem. Polymerase doesn't like being heated. If I heat the sample to separate the strand, polymerase doesn't work. So it's like crud. Like I have a solution, but now my solution to one problem causes a new problem. So Kary Mullis, the guy who invented this, I'm not sure if he didn't discover this, but he used an enzyme called Taq polymerase, TAQ. Taq polymerase. So there are bacteria that live in like hot springs. Thermophilic bacteria, like around the Old Faithful or whatever, those bacteria live in very hot places. So their polymerase must like it hot. Taq polymerase is the kind of polymerase, do I have this on here? No, I don't. And made by thermophilic bacteria. So we don't use human polymerase, we use Taq polymerase in PCR because it doesn't mind being hot, all right? These next couple of slides, this is just interesting. So Carolina Biological is a company that supplies science supplies for schools. PCR, like I did PCR in college. PCR, you wouldn't do this in high school. You might do it in high school, but this is more of a college thing. You can buy what's called a thermal cycler. It's over $5,000, which that's a lot of money. But in terms of a laboratory piece of equipment there's things in colleges that cost $1 million, like big old NMR machines or whatever. This is a little tabletop. It's basically an oven. It's like a thermal cycler. Like it heats up, cools down, heats up, cools down. Some high schools do have these, but again, that's a lot of money for something to do in high school. And you can also buy from Carolina these PCR beads and read this. It says, save time in your classroom and lab with these PCR beads that are pre-formulated, pre-dispensed, and include all reagents required for PCR reactions. Simply add your DNA template, water, and primers, which the DNA you have, water you have, primers, you can order primers. Less pipetting means less contamination and fewer errors, yielding robust, reproducible results. Use these beads with confidence to replenish your Carolina PCR kits or for other PCR reactions. And 25 of these beads is 70, well, the beads are in these little Eppendorf tubes. So 25 of these is $75, which, I mean, 10, 15 years ago, this would be a fortune to do this. Now you spend 5 or 6 grand on a thermal cycler, you buy these beads, which aren't that expensive. And it's funny, here, see how it says available November 16th, 2018. I guess I screenshotted this in 2018. So this was so new that it wasn't even available yet. It was going to be available. We should see if it's available now. I'm sure that it is. But you know, this being more like, again, you could do this in high school. 10, 15 years ago, you're doing this only in college or in research institutions. So these are tools. These are biotech tools that we use to do genetic engineering. This slide just shows, I'm not quite sure why. So I guess the idea is you use PCR to make the fragment, make copies of it, then cut it into the plasmid. Here's a recombinant plasmid. Put this plasmid into a bacteria. The bacteria have been engineered. The last thing that I want to say is a word on DNA sequencing. So DNA sequencing is a way of taking a DNA strand and figuring out what the sequence is. ATG, CTC, ATCT, ATG. What is the sequence? If you don't know the sequence, you had to know the sequence to do stuff. When I was in high school, there was a worldwide project called the Human Genome Project. It took 15 or 20 years. Actually, I'm not sure. It took over a decade for sure. And the goal was to sequence the entire human genome. And I think it was finished in 2001. And it took labs across the world, again, over a decade. Now you can sequence DNA overnight. Like the technology that we use to sequence DNA is so fast and it's automated. There's hospitals, a kind of new thing you can do is, you know, say a baby is born with a disease. It's a gosh, 1 hour old baby, and you can tell it has a disease. How do you diagnose that? Like the baby can't tell you anything. What if I could take their DNA, sequence it, and see if I can diagnose the disease based upon their DNA? And some hospitals do this, and you can basically get the sequence overnight, maybe 48 hours. But you know, take the sequence, put it to an automated big computer thing. It sequences it, spits out the sequence. A computer looks for DNA mutations, and you can sequence diseases by finding the mistake in the DNA. That's huge. You know, 20 years ago, that would have taken, I mean, it would have been so expensive and taken, you know, months or years to do that. That's not practical. Right now, you can do it virtually overnight because the technology is so good. Okay, so that was a lot in one chapter. Hope that was helpful, and I will see you guys next time.